Thank you so much for joining us in this webinar. We are very happy to have this opportunity to discuss wound healing assay with you. So I'm gonna share my screen with the presentation. Just a second here. So, all right. I hope everyone can see the slides. Is that okay, Patricia? Okay, perfect. So today, as I was saying, we are going to talk about wound healing assays. So my name is Katja. I'm the application scientist at 4DCell, and I will be one of today's presenters together with my colleague Patricia. Just a little background on us. So I have a PhD in biomedical sciences, and my main background is immunology and biochemistry. And Patricia, she's the R&D project manager at 4DCell, and she has a PhD in biomaterials, and her main area of expertise is microfluidics. Before starting with the presentation, I want just to tell you that the webinar is divided into parts. So first we have the presentation itself, which is then followed by a question and answer session. You can already during the presentation type your question and send to us, and we will read it in the end of the presentation. Otherwise, you can also then in the end either type your question or raise your hand, and we will have you give you access to the microphone. So 4DCell, for those who don't know us, it's a biotech located in Paris, in France. We develop in vitro tools to control the cell microenvironment. And our idea is to provide bioengineered in vitro assays for academics and companies to in order for them to acquire more relevant and accurate readouts from the experiments, in vitro experiments performed with cells. And to your right, you can see here our facilities where we manufacture the products we send to our clients. So before going directly to the wound healing assay, we thought it would be nice to just give you a brief overview of the wound healing process, because it's quite complicated and it involves many molecular mechanisms. So the wound healing, it's I'm sorry. the wound healing process, as I was saying, it's a complicated process that involves many different cell types that coordinate among themselves different functions during the different stages of wound healing. And the wound healing, it happens all the time in the body, right? So, for example, when you cut your finger, but it's also involved very much in different pathologies, such as diabetes and also lung related disorders with the scarring of the lung. So taking this into account, we understand that there is a huge need for good in vitro models, particularly that allow the use of high throughput screening platforms, for example, if developing or screening a library of drug leads when the therapies are under development. So regarding wound healing itself, it's divided in 5 main stages. The first stage is hemostasis, where the blood vessels start constricting and the platelets are activated to form a fibrin clot, which then prevents the blood flow, or basically it stops the blood flow. This creates a sort of a scaffold for immune cells to be able to create a line of defense against microorganisms such as bacteria and viruses. And this stage of inflammation, so the second stage of the wound healing process, the immune cells are also important for clearance of cellular debris and also for resolution of infection, if that is the case. So this leads us to the third main step of this process, which is angiogenesis. Basically, what happens here is that the endothelial cells start proliferating and migrating to the injured area to form new vessels. Following this, there are resident fibroblasts, which differentiate in myofibroblasts and also migrate to the injured area. And what they do is to constitute or to help forming the contractile tissue and also creating a specific microenvironment that will help repairing the epidermal layer, which leads us to the last stage of the wound healing, that is basically tissue remodeling and ultimately the closure of the wound. So we are going to show you our in vitro assay to study wound healing, and you can think about that it can be used at different stages if you are studying one or the other, and it can be different cell types can be employed in this assay. But we can also discuss this later on during the question and answer session. So I'm going to give you a little bit of background on the technology that we use to develop this assay. So the technology is based on micropatterns, which are substrates chemically modified in a way that to have cells attaching to a glass, for example, only in specific areas. As you can see here, the cells are attaching to these lines or rectangles, white ones, whereas there is a non-adhesive coating surrounding the adherent areas, which do not allow the cells to attach outside the adherent areas. So the cells in these areas can attach either to bare glass, or you can coat it with extracellular matrix protein, for example, fibronectin. There are different shapes available for these micropatterns, and this depends a lot on the type of cells you are working with, what kind of readouts you are intending to obtain. For example, there are micropatterns of line shapes, squares, triangles, or we can also customize a different design if there is some specific request. This is a cross-section of a micropattern cell on a glass cover slip, so you can see like from another perspective. So what I just explained is how the standard micropatterns work. So the principle is similar for the wound healing assay, but we use something called click chemistry-based polymer. And for the wound healing, we specifically call the micropatterns dynamic micropatterns. So I'm going to explain to you how the method works, or how do we develop this method. So basically what we have is a glass cover slip, for example, coated with a non-adhesive polymer, which then we can print with a specific pattern for wound healing, a specific design that we made for wound healing. So once you proceed with the patterning process, there are areas of the glass that will become non-adherent, that will become adherent after the patterning process, which leads to this kind of feature. As you can see, there is an area which is still non-adhesive at this initial stage, where the cells cannot yet bind or attach. Meanwhile, there is new areas after this patterning process where the cells will be able to attach, as you can see here, basically surrounding this wound area or this wound stripe. And at some point after seeding the cells, you can add a trigger protein, which is an analog of fibronectin. And what this protein does is to induce or to render this non-adhesive area to become adherent, which then allows the cells that were in these sites to start moving into the wound area. So at this point, all the surface of this cover slip is adherent. So you can see the cells starting to move on top of basically in the wound area. So this is more or less the theory behind how the dynamic micropatterns can be used to prepare this wound assay. And Patricia will give you specific examples of experiments that we have done, and that will illustrate better and make it easier to see how does this work. So these dynamic micropatterns for the wound healing are provided, for example, in glass cover slips, as I give you the example, also in glass bottom Petri dishes, but as well in multi-well plates of 96 and 384 wells. And this format with the multi-well plates is very good and very convenient if wanting to, for example, screen a large library of compounds, or if you want to compare many different conditions at the same time. So now I'm going to pass the word to Patricia, who is going to give you some specific examples. Patricia?
Yep, thanks, Katja. So Katja explained to you very nicely how we make the micropatterns. And so here I'm showing an image of mouse embryonic fibroblasts that I patterned. And so in the center, you see a 500 micron wound. And you can see that the cells are very well only on the micropatterns, and you have a very smooth border. And so if you add the trigger protein, you can see that the cells lose the smooth border and they start migrating into the wound very nicely. So in this case, like I said, I'm using mouse embryonic fibroblasts, and we're using a concentration of 25 micromolars. And I left the trigger protein in the supernatant. So we can think of various things we can test. For example, whether we can use a similar assay with other cell types, whether we can use a different concentration of the trigger protein, and whether we can vary the amount of time that the trigger protein is left in the supernatant in case it affects the migration of the cells. So next slide. So the first thing to test is whether we can do this assay with other cell types. So here I am showing 5 cell types that we've tested. So we have human fibroblasts, mesenchymal stem cells, HeLa cells, mouse embryonic fibroblasts, which I just showed, and mouse myoblasts. And you can see that all of these cell types pattern very nicely. And in fact, once you add the trigger protein, the cells will migrate into the wound at various speeds depending on the specific cell type. So the next thing we tested was whether we can change the concentration of the trigger protein. So here you could see I tried 5, 15, and 25 micromolar, and I was actually surprised that I didn't see any difference in the migration speed. So you can see the wound is closing pretty much at the same speed for the 3 concentrations. So I tried a concentration that was even lower, and in this case, for 1 micromolar, the mouse embryonic fibroblasts were not able to migrate into the wound even after 24 hours. I also tested human fibroblasts, which did not migrate either, and C2C12s. And for the C2C12s, surprisingly, we could see that there's some cells that are able to migrate into the wound, but they don't migrate as a united front as you'd expect for a classical wound healing experiment. So the minimal concentration is really cell type dependent. And if you want to test out this assay, I would suggest testing different concentrations. In our case, we provide the amount of reagent that you would need to do experiments with 25 micromolars. So we think that this difference in the concentration is happening because when you use 25 micromolars, you're really saturating the surface so the cells are able to adhere all over the surface. But if you use a much lower concentration, such as 1 micromolar, you reduce the amount of attachment points that the cells have because you're adding fewer trigger proteins. And so this can allow migration for cells that need fewer attachment points. But this could also be an interesting assay for you if you have a particular application in mind that in which you'd like to modulate how well the cells can attach. And so if this is something interesting for you, we'd actually like to hear about it. And so another thing that I wanted to test was whether we could reduce the activation time, so the amount of time that the trigger protein is in contact with the cells. So what we do first is we replace the medium with medium containing the trigger protein. We incubate it for a specific amount of time. So in my experiments, I tested from 1 to 30 minutes. And then you remove the trigger protein and you replace with complete medium. And when my experiments with mouse myoblasts, and I'm showing also with human fibroblasts, I saw that the cells migrate into the wound. And migrate quite nicely. In all experiments that I did, even as short as 1 minute was sufficient to have a complete closure of the wound. And so with all of these experiments, we can also get very nice movies, such as the one I show here, where in this case, I'm using a slightly larger wound, 1 millimeter, so 1,000 microns. And with the mouse embryonic fibroblast, it takes about 48 hours for the wound to close. So we can get very nice data. As I mentioned, we can try different sizes of the wound. So in this case, we're showing 500 and 1,000, but we can also try different sizes. And in fact, you could imagine a variety of shapes. So in this case, we're showing several wounds in parallel. So these are quite small, 100 microns, but the advantage of this is that you can see several in your field of view. And we can even imagine having a completely different arrangement. So in this case, I'm showing a circular shape that's 500 microns, and you can watch how the cells migrate out of this. And as you can see from the bottom, I'm showing experiments using different cell types. And so here on the left, we have HeLa cells, and we also have mesenchymal stem cells. So this assay really works with a lot of cell types. And so now I'm going to pass it over to Katja, who will tell you about more advanced applications.
Yes, exactly. So we thought it would be cool to discuss some more specific applications that we could do, or variations of how to use the dynamic micropatterns to study cell migration as well, but wound healing in this case. So we thought about combining, for example, the dynamic micropatterns with the cell confiner, which is an instrument that we have at 4D-Cell. And the principle of the cell confiner is basically to have cells compressed between two parallel surfaces to a specific height, which you can choose from the beginning. And the idea is to have the cells, for example, attached to a glass cover slip, let's say, or a petri dish or a plate. And then you basically have assemble a silicone piston that is attached to a glass cover slip with the micropillars of this specific height that I said. And then you close the system, and the cells, as I said, will be confined or compressed between these two surfaces to the height that you predetermined from the beginning. Usually researchers use between 1 to 20 microns of height, of confinement height. And we thought about this, the cell confiner for the wound healing, because, for example, if you're interested in studying at the single cell level, because the confiner is really good in that sense, because I'm sorry, the cells, you can get very good images and live videos of cells at a very close level, in the sense, for example, to study events taking place at the membrane, the cellular membrane, but also to look at how the organelles move, if something happens there, and also for nuclear deformation and cell migration itself. So when combining this with the dynamic micropatterns, it's very similar. Instead of having just the cells attached to the bottom of a petri dish, for example, you can have the cells already in the dynamic micropatterns seeded, and then you add the trigger protein, which will make then the non-adherent area to become adhesive. And then you close the system, the confiner, and you can see here that the cells will be compressed to the height that you predetermined. And this will allow you to do these additional experiments that I was describing. We have here an example, for example, of bone marrow dendritic cells that once under confinement, they change their phenotype and their features when they migrate. For example, you can see that they acquire or they protrude in these lamellipodia. And this video also illustrates that. It's really cool. You see the cells here projecting the lamellipodia on the sides. This is just the same cell type, but with a mutation. So by combining the dynamic micropatterns with the confiner, it's possible to assess this type of studies if this would be something you are interested in, not only the collective cell migration towards the wound healing. And now Patricia will give you just one more example. Is the same, but the main difference is that the dynamic confiner allows you to see the moment you are confining the cells, because it is connected to a vacuum pump. But they work similarly anyway. And if you have any questions, then we can later on discuss how it works. So yes, now I pass to Patricia.
Okay, thanks, Katja. So I just wanted to briefly mention one of the products that we're developing right now. So we are developing a way to cut silicone membranes very precisely. So here you can see that I'm mimicking a 96 well plate by cutting out a membrane and then adding it onto a glass plate. And so using this, we could imagine that we could do lots of different things. For example, you could have a Petri dish in which you have several wounds, and you could use this type of stencil that we would cut for you to have different areas in the Petri dish where you may have, for example, 2 cell types, or you want to look at the same cell type, but using different media on, or different treatments on in the 2 sort of wells. So yes, so this is something we're developing right now. And if you have interest in this type of product, of course, you can contact us. And so thanks for your attention. We're really happy to be able to share this information with you. Our next webinar will be on nuclear mechanics and deformation after we take a break for the summer.